And I'd like to introduce Dr. Dong-Yoon Kim before she shares her opening remarks. Dr. Kim is a mathematical statistician at the Office of Biostatistics Research within National Heart, Lung, and Blood Institute. She received her PhD in statistics from the University of Michigan in 2003. And before joining NIH in 2013, she held a faculty position at Virginia Tech. Dr. Kim has years of experience in collaborative research in other areas, including mobile health, bioengineering, and environmental science. She's also serving as the president of the Caucus for Women in Statistics and a board member for the Korean International Statistics Society. Please join me in welcoming Dr. Kim, who will now give her opening remarks.
Thank you very much, Dr. Kim. Good morning to Korean audience and good evening to the US audience. Welcome to the fifth installment of GISTeR-CWS webinar series. This international event, Bridging the Gap, Advancing Research Excellence Through Equity, Diversity, and Inclusion, is co-hosted by the Korea Science and Technology Gendered Innovation Center, GISTER, and the Caucus for Women in Statistics and Data Science, CWS. This series was established last year with the shared goal of bringing critical attention to pivotal topics in women's health research through the perspectives of experts from around the world. Dr. Hae-suk Lee, Director of GISTER, and I, past president of CWS, are honored to co-chair this series, working together to provide a platform that fosters dialogue, insight, and collaboration in a global scale. Each session has featured outstanding speakers who have advanced our understanding of health disparities, biomedical research, and gendered innovations. Let me briefly talk about the organizing committee in the US side. The committee consists of dedicated CWS members. All of them happen to be within the NIH community. As such, we have been privileged to host several NIH experts as invited speakers. One of the first speakers was Dr. Jill Barhart Sloan from the National Cancer Institute, and most recently, we had the honor of hosting Dr. Vivian Ora Wang, Deputy Director of the Office of Research on Women's Health at NIH. Today, or tonight, depending on where you are, we are pleased to continue the tradition of impactful discussion and insight by inviting two distinguished speakers from US and South Korea. We hope that this evening's presentations and discussions will not only inform, but also inspire further collaborations and understanding on why sex and gender matter in women's health research. Thank you for joining us, and we look forward to a meaningful session ahead. Thank you.
Thank you, Dr. Kim. Now I'd like to introduce our first speaker, Dr. Jong Park, a faculty member in Department of Biophysics at Sungkyunkwan University and adjunct professor in the Department of the Metabiohealth at Sungkyunkwan University. Dr. Park has received his PhD in biomedical science from Seoul National University, and his research focuses on the therapeutic approaches and drug development using iPSC-derived organoids. Also, as well as the neurodegenerative disease such as Alzheimer and Parkinson's disease. Dr. Park is also interested in identification of the biofluidic biomarkers for early diagnosis of dementia. Today, he will discuss sex differences in Alzheimer's disease and the clues from the brain metabolism. Sex differences in Alzheimer's disease have becoming a significant area of research, revealing important distinction in prevalence, pathology, and symptoms and response to the treatment. So please join me in welcoming Dr. Park as he presents his work on this topic.
Thanks for a kind introduction and thanks for having me today. I'm really honored to speak here about my research topics. So as my title is, today's topic title is Sex Differences in Alzheimer's Disease, Clues from Brain Metabolism. So actually I'm doing work about the brain organoid and clinical neuroscience as well. So today I introduce about the blood-based biomarker and about the brain imaging data to identify the sex differences in Alzheimer's disease related to the brain metabolism. So before I talk about the real research data, I want to introduce about the definition of sex and gender. So sex means that sex refers to the biological characteristics, the primary and secondary, that differentiate females from males. So if we want to talk about the real biological meaning, then we need to use the sex instead of the gender. But however, the gender refers to the socially determined meanings of being a man or woman, which shapes the definition of feminine and masculine behaviors, products, or technologies, or environment, and so on. So today I'm going to focus on the biological characteristics of the between the females and males. So I will use the word sex and females and male instead of the gender word woman and man. So please kind of understand about these words about the in the research field. So before I talk about the sex differences, I'd like to introduce what is the Alzheimer's disease. So maybe the many of researchers already know about what is Alzheimer's disease. So this is the major dementia type in the world. So there are two major pathological hallmarks. The first one is amyloid beta protein, and second one is extracellular tau deposition. So these two major hallmarks is the main pathologies, but there are many environmental factors and multifactorial factors to inhibit or enhancing the pathology of the Alzheimer's disease. So currently, there is some rising concerns of the drugs. So as you can see here, currently in Korean FDA, one of the drug called Lecanemab, the name is actually lecanemab, is approved by the Korean FDA. But actually currently there are rising concern about the sex differences. So as you can see here, many researchers have concern about the lecanemab doesn't appear to work in women. So there are many differences about the efficiency of the drug, and the men have much higher efficiency than rather than women. So many researchers are trying to find why it is occurred and what is the problem of the sex difference between the male and female. So as you can see here, the amyloid beta and tau protein is the major pathological hallmarks. And the first amyloid beta occurred like this. And after that, tau pathology and clinical dysfunction can follow after the amyloid beta plaque deposition. However, there is a small differences in amyloid beta burden, but it could become amplified in downstream processes such as tau and clinical dysfunction. So although there is a small difference, but when the disease goes through, the amyloid tau protein and the clinical dysfunction can be very different like this. So we need to focus on why this can happen between the male and female. So many research papers are showing that the difference between the male and female in the progress of Alzheimer's disease. As you can see here, depending on the age progression, there can be the difference occurrence of the pathology between the male and female. So generally, the female have much higher proportion of the dementia rather than male. And also about the genetic risk factor such as ApoE, you can see there are a big difference tendency between the male and female like this. So as you can see here, the ApoE4 is the most popular risk factor of Alzheimer's disease. The female didn't have any the different tendency between the ApoE type, but male have different tendency between the ApoE4 risk factor and non-having risk factors. So as you can see here, both the aging factor and the genetic risk factor, there is a big sex differences in the progression of Alzheimer's disease. So let me introduce more about the sex differences in Alzheimer's disease. So as you can see, this left side, there's some difference between brain atrophy. So this is the a figure between the females and males. So as you can see here, the females have much faster atrophy was observed in females than men. And also, maybe you already know about the difference of the sex hormone. So the sex hormone difference can also make difference between the female and male. And next thing that can happen is chronic stress. So as you can see the left side, there's a sex difference in chronic stress responses make Alzheimer's disease, especially about the Krebs cycle and the ERK pathway. So although there's a similar chronic stress, there can happen the different responses between the females and males. So it also can make a difference between the sexes. And currently, the many researchers are focusing on the metabolic reprogramming differences between the sexes. So as you can see here, the center region is about the window of treatment opportunity for the disease prevention and disease retardation. So this is a very important period about the Alzheimer's disease progression. But in this case, the female normally can have the big differences level of the sex hormone. So many researchers are saying that this phenomenon can occur, the sex differences between the females and males. So let me more focus on the glucose metabolism in Alzheimer's disease. So maybe many researchers know about the Warburg effect. The Warburg effect is the phenomenon in which cancer cells or the dying cell can produce the additional energy through the increased oxygen-dependent glycolysis, followed by the lactic acid fermentation with secretion of lactate. So this is the Warburg effect. And many researchers have evidence that this Warburg effect and hypermetabolism of Alzheimer's disease in AD patients or AD mouse models can make some difference between the sexes. So in Alzheimer's disease, especially about the microglia cell, there is a different mechanism between the healthy brain and Alzheimer's disease brain, or some Parkinson's disease brain. So as you can see here, in the healthy brain, there is an OXPHOS system. This OXPHOS system can make an ATP in the normal status. However, when there is some acute disease status, there is some increase of the glycolysis levels. And after the acute status, there can be the chronic status. And at this stage, there is some decrease of OXPHOS system and decrease of glycolysis system. So this kind of metabolism is highly associated with the Alzheimer's disease progression. So if there is some difference between the sexes about this progression of the metabolism, then it can have the difference between the sexes. So recently, there is some interesting paper about the sex difference in Alzheimer's disease about metabolic reprogramming and the therapeutic intervention. So this figure shows the bioenergetic defect between the female and males. So as you can see here, there's some different tendency of the bioenergy to defect between the sexes. And also, I already showed this graph. This metabolic state is also very important the phenomenon in the window of treatment opportunity for disease prevention and disease retardation. And at this stage, there are some sex hormone differences between the males and females, and maybe it can occur the different result of the metabolic states in Alzheimer's disease. So this table summarizes about the hormone changes and changes in brain glucose metabolism and also the potential link with Alzheimer's disease. So these hormone changes can increase in sex. These hormone changes can increase about the risk of the Alzheimer's disease, especially about the increase in brain glucose uptake during the puberty. And also it can make some difference risk about the Alzheimer's disease. So maybe you can see this table, and the main point of this table is the changes in sex hormone levels and glucose metabolism throughout the reproductive life of women can make some difference between the male and female. So my research are trying to focus on these aims. The first one is to investigate direct evidence of link between the sex and glucose metabolism. And aim two is to find the blood-based biomarkers, the well-known biomarkers for glucose hyper and hypometabolism in Alzheimer's disease. And third one is to identify the candidate genes linked with the glucose metabolism in Alzheimer's disease. And fourth one is to identify the discrimination power of these biomarkers, blood-based biomarkers for glucose metabolism. So these two slides are showing the methods of my research work. So first, I measure the plasma biomarkers. The first one is total tau plasma biomarkers. I quantify the total tau levels with the Simoa kit. It is developed by the Quanterix. On the HD1 analyzer. And next one is plasma amyloid beta biomarkers, such as amyloid beta 42 and amyloid beta 40, which is the major, the general biomarker of Alzheimer's disease in the blood. So I quantify the plasma amyloid beta levels by Innovia plasma amyloid beta forms kit by using the Bioplex 200 system. And also to know the genetic vector of the sex differences in Alzheimer's disease, I performed the transcriptomal analysis using the publicly available database. And also I performed the functional network modeling by using the string database as well. So I used the two cohort, the cohort 2 and 3, and the publicly available database name is the GEO database containing the human brain tissue samples and the prefrontal cortex samples, and the number of patients is 78 samples and 18 samples. And also, I performed the protein-protein interaction network and functional enrichment analysis by using the string database and by using the DGs from RNA sequencing data. About the statistical methods, I performed the monotone regression spline curve analysis to mimic the disease progression of Alzheimer's disease. And also I performed the RC curve analysis to know the discrimination power of the biomarkers. And also to integrate the biomarkers, I performed the logistic regression analysis and multiple regression analysis. And also these data can include the brain imaging data such as FDG PET. The FDG PET is about the quantifying the glucose metabolism levels and the neurodegenerative levels. And also we have the PIB PET imaging data. It can represent the amyloid beta levels in the brain. And also about the cortical thickness and hippocampal volumes by using the MRI data as well. And to know the correlation efficiency, I performed the partial correlation analysis with the correction of the covariance, such as the education level or the aging, and also the chi-square test for the proportion of the sex, the proportion of the female and males. And also I performed a randomized selection to match the number of patients between the male and female, and also the comparison of slope analysis as well. And to visualize the data, I performed the medical software and rapid precision aid and other the statistical tools for statistical analysis. So this is the experimental design. For cohort 1, I performed the I quantified the blood-based biomarker levels, both the baseline and follow-up cohort. So the data includes the plasma amyloid beta 42 and 40 and ratio, and also the tau levels. And also we have the brain imaging data. By using these imaging data and the blood-based biomarker data, we perform the longitudinal analysis, and also we perform the supply curve analysis and partial analysis and R-scope analysis to reveal the female-specific glucose metabolic dysfunction and its reflection on the AD plasma biomarkers. So this cohort 1 is for the blood-based biomarker studies, and cohort 2 is slightly independently. So the cohort 2 is for the for identifying the female-specific genetic factors which related to the glucose metabolism. So by using the GEO database, we performed the transcript analysis and performed the string PPI analysis and GEO gene ontology analysis and R-scope analysis as well. So by using this human brain data, so we revealed the female-specific glucose metabolic genes in Alzheimer's disease, and we validated this result by using the cohort 3 with the 18 participants. So this is the demographic data of our cohort. So we use the K-based cohort, which is derived from the Seoul National University Hospital by Professor Dong-Young Lee. And also we use the public database analysis. So first, we need to check the sex differences in the brain imaging data before we start the analysis for the blood-based biomarker. So as you can see here, the tendency about the progression of Alzheimer's disease is totally different between the male and female. So this monotone regression spline curve analysis can show the disease progression versus the brain imaging data. So the male can have some fluctuation, but female can have directly the correlation between the amyloid beta progression and the other the brain imaging data. So as you can see here, when you compare the spline curve between the male and female, so you can see there are big difference and significant level of p-value like this. So we confirm that the fact that male and female have different tendency of the progression of Alzheimer's disease, and so we start to analyze this data for the longitudinal analysis. So we confirm that the female have difference the glucose metabolism levels between the Alzheimer's disease patient and the normal participants. And also this figure D showed that the Alzheimer's disease patients have much higher proportion of the females which have the severe glucose metabolism levels. So it means that although there is some similar Alzheimer's disease patients between the female and males, the female can have much higher proportion of the glucose metabolism deficiency. So we compare the amyloid beta level and the glucose metabolism level. And very interestingly, the female only have the significant correlation between the amyloid beta level and the glucose metabolism. And after we cracked the number of the subject between the male and female, because actually Revere will comment about the difference between the number of patients. So we performed the randomized selection for females, and we matched the number of patients between the males and females. So although we corrected the number of patients, we still can see the significant correlation between the glucose metabolism level and the amyloid beta deposition levels. So it shows that there is some the sex differences in the brain hypometabolism in patients with Alzheimer's disease. So we performed the monotone spline curve analysis as well. So as you can see here, the amyloid beta levels have correlation in the female patients. So we thought that this amyloid beta 40-40 ratio can represent the glucose metabolism dysfunction in Alzheimer's disease. So very interestingly, the plasma amyloid beta ratio have significant correlation in female, but not in males. So it means that the plasma amyloid beta 40-40 ratio can represent the disease progression about the glucose metabolism level in female, but not in males. So it means that the plasma biomarker also can have different tendency between the males and females. So so far, we found that the big difference of the brain imaging data between the males and female, and also big difference about the plasma biomarkers between the males and females. And we move on to the brain data to reveal the female-specific genes about the glucose metabolism in Alzheimer's disease. So we collect the GISTeR, the publicly available data. So it includes the 78 participants, and it has a similar number of patients, as you can see here. And also we can check the UMAP data. So you can see there's some cluster of the control data and Alzheimer's disease data. It shows a very clear cluster result from the UMAP data. And very interestingly, although the number of patients are very similar between the male and female, the female only have very many differentially expressed gene between the Alzheimer's disease and cognitively normal participants. So although the Alzheimer's disease, the main Alzheimer's disease patients are the 22%, and the female Alzheimer's disease patient are 24%. It is very similar number, but the RNA sequencing data show that the big difference number of upregulated and downregulated genes. So it was very, very interesting. So we tried to find the much details about the differentially expressed genes. So we selected the glucose metabolic genes from the Reactome database. It is the very popular, the publicly available database to know the disease-associated gene or disease-associated pathways. So from the Reactome database, we collect the metabolic-related genes, 2,208 genes. And also we collect the glucose metabolic genes. It is the 92 genes. And this is the genes from the Reactome database. And also, we have the differentially expressed gene from this publicly available database of RNA sequencing data. So we tried to compare this RNA sequencing data versus the Reactome database. So first, we check the metabolic genes, which is overlapped with our RNA sequencing data. but we couldn't find no big difference between the proportion of the metabolic genes between the males and females. So although the number of genes are significantly different, but when you see the per milli, the percent per milli is the 8.7 and 8.4. So we cannot see any difference between the male and female about the metabolic genes. However, when we focus on the glucose metabolic genes, so as you can see here, there are big difference of the number of genes, and also there are big difference of the percent per miles per millis between the males and female. So approximately three multiple genes are the difference between the in the female genes. So there are 26 female-specific glucose metabolic genes, but there are just one genes about the male-specific brain metabolic genes. Okay, so when you see there, however, there is one genes overlapped between the male and female. The one gene is the NUP18A. So I excluded these genes for the to make the female specific gene list. So as you can see here, there is a 19 genes about the down-regulated genes, and 20 genes of down-regulated genes, and there are the 5 unregulated genes in females with Alzheimer's disease. So these genes mean that there are specific and the female-specific genes about the glucose metabolic dysfunction. It was a very interesting result. So we narrowed down the female-specific glucose metabolic genes from the RNA sequencing data and the reactome data. Then we need to find the most important gene, which is related to the Alzheimer's disease. So I performed the string protein-protein interaction analysis by using string database. So as you can see here, I selected these 25 genes from this data, and I performed the PPA analysis with the Alzheimer's disease relevant genes. So as you can see here, the APP and MAPT and base 1 and 2 is very famous and very general risk gene of Alzheimer's disease. And I put the genes as well with the glucose metabolic genes. And I tried to find the directly linked genes with the Alzheimer's disease genes. So very interestingly, we found the 4 genes are directly interact with the amyloid beta genes and the tau-related genes. The 4 genes are ARDO-A and INO2 and PRKACB and PPP2R50. So I confirmed the expression level of these 4 genes. Then as you can see here, the ARDOA and the PRKACB and INO2 and PPP2R5D is only have significance in females, but we cannot see any significant differences in the males between the control and Alzheimer's disease patient. So it means that these 4 genes are highly associated with the Alzheimer's disease, but not in the male patient, because it has only significant levels in the female patient between the control and Alzheimer's disease. So to know the diagnostic power of these 4 genes, I performed the logistic regression analysis to integrate these 4 genes to perform the RC-curve analysis. So after I integrated these 4 genes, and I performed the RC-curve analysis and confirmed that the AUC value, the AUC is the area under curve. It means that the diagnosis power of the disease. So I compared the contour and Alzheimer's disease patient for both male and female. But as you can see here, the female have the significant level of the RC curve model fit, and also it has the higher diagnostic power of AUC. The power is the value is the 0.880. However, we cannot see the significant level in the female patient, and also it has the less power of the AUC rather than the females. So it means that these four genes can be used for diagnosis of Alzheimer's disease in female, but it is not appropriate for the male patients. So we thought that this is a very interesting result because some the current drugs does not have any effect for the male patient. So maybe this kind of phenomenon can have some effect about the difference effect of the drugs. So this is the brief summary of the analysis of this paper. So we use the blood-based biomarker for the longitudinal analysis, and we confirmed that the plasma amyloid beta levels ratio can represent the difference of the brain glucose metabolism level in Alzheimer's disease. And for the brain, we use the transcriptome data, and we confirm that there's four female-specific glucose metabolic genes, and it can directly with the Alzheimer's disease genes, and also it has some very high diagnostic power to discriminate the contour and Alzheimer's disease. So of course, this is supplementary data. We also perform the plasma. We also perform the RSCAV analysis by using the plasma amyloid beta 40/40 ratio. It also shows the much higher diagnostic power in the female rather than males. So this also can say that the plasma amyloid beta 40/40 ratio levels also can be used for the screening and predict the Alzheimer's disease in female, but not in the male patient. So this is the summary of the results. So in conclusion, our longitudinal, the blood-based biomarker data and the brain transcriptomic data can clearly reveal the sex differences in the human brain glucose metabolic changes. And next one is by using the 2-year follow-up cohort, we also showed that the brain metabolic impairment was revealed only in women, and the plasma beta amyloid 40/40 ratio was a possible biomarker for brain metabolism in women. And also we identified 25 novel female-specific glucose metabolic genes. And also we narrowed down the key attractor genes. We can call them female-specific glucose metabolic genes for Alzheimer's disease, and we narrowed down to the 4 genes, ARDOA and INO2 and PRKACB and PPP2R5D that was associated with the amyloid and tau related genes such as APP and MAPT and BACE1 and BACE2. So from this result, we can offer the crucial evidence on sex differences in pathological Alzheimer's disease mechanism. And we can suggest that these differences can help to elucidate the pathophysiology of Alzheimer's disease, and maybe it can have a clue to develop the appropriate diagnostic or therapeutic treatment for Alzheimer's disease. So these results so far is about the blood-based biomarkers and the clinical application of Alzheimer's disease. But as I mentioned, that my different field of research is about the brain organoids using the patient-derived iPSCs. So I currently developed the multi-region brain organoids and mechanism pathways, and also trying to find the disease clue of Alzheimer's disease. So I'm trying to focus on the sex differences as well by using the brain organoid, because we can make some patient-derived iPSC cells. Maybe it can have the difference about the cell metabolism or the pathophysiology of Alzheimer's disease, although it is derived from the iPSC cell. So maybe we can check the difference between the female-derived iPSC cell and male-derived iPSC cell as well. So I'm going to try to find some difference between the sexes by using these different models. Okay, so about today's talk, the main portion of the works are done in the Professor Jong labs when I'm doing the postdoc. So for this project, the student Han-Byung Lim and Dr. Son Han was the very big help for me, and also many patient samples and blood samples and imaging data are from the Seoul National University Hospital by the Professor Dong-Young Lee laboratory, and also the Professor Min Soo-byun and Dae Lee and the Gi-Wan-byun and Gi-Jung Jeon and Professor Yu-Kyung Kim also help this study. And also about the collaborator, the postdoc in KAIST and Sungkyunkwan University and University College London and Harvard Medical School. I really thanks to every researchers. And this is my laboratory, and I really thank my lab members. And this is the funding disclosed. And thank you very much for today's talk.
Before we go to the Q&A session, I'd like to make a quick announcement. We have a survey link for the improving, on awareness of the gendered innovation in the chat. So we really value your input and feedback. So we invite you to fill out the survey and please take a moment to share your opinions. Can you see the links in the survey? Yes, I can see the links in the survey. So I think one is in Korean and one is in English. So please take a moment. So now I'd like to open the floor for the questions. Do you have any questions from the audience? As more people live longer, I think we might see increased prevalence of Alzheimer's disease and changes in healthcare. And research continues to evolve, aiming to uncover the complex interactions between brain metabolism and Alzheimer's disease to inform better treatment and prevention strategies. I was just wondering, this public database, the GEO database you mentioned, what other variables would be available in this public data? Like, for example, do they have a history of medication, like medicine, medication, like?
Actually, there are not many other factors that can be used because they focus on the RNA sequencing data, and they only give us the information about the sex and age, and not many information about the demographic data. It is actually kind of limitation. Yeah.
I see. Yeah, I think sex is a biological factor in Alzheimer's disease is important. Right. Understanding sex differences can help us identify who is at higher risk for developing the disease, and thus enabling more targeted prevention efforts and more tailored treatment plans.
Right.
So yeah, so I was just wondering, how the effects of the medication could change or moderate the sex differences.
Yeah.
Thank you.
Thank you. Yeah. So one thing I can let you know is there are many public RNA sequence data, and although this data is just show us not many the demographic data, but some public data have very many the demographic data, clinical data. So maybe many researchers can do the very interesting the analysis by using the GEO database. It is totally free, and we don't have to get permission from the authors. So it is very useful to many researchers. So I strongly recommend to use the public database as well.
Thank
Great,
you. I
thank you.
have a question.
Yes, please.
Thank you very much for your great talk. And then in what ways can treatment for Alzheimer's be tailored based on sex difference, and what role might personalized medicine plays?
Sorry, can what wait?
Oh, actually, the question is in the chat.
Yeah. Yeah. So I think as I told today, so maybe we need to find some female specific mechanism so it can go to the personalized medicine for female, because actually the current drugs such as donanemab or lecanemab is just targeting the amyloid beta inside of the brain by using the antibody therapy, but it did not consider the specific mechanism of Alzheimer's disease. So actually, today's talk is one of the clue and one of the approaches to target the specific mechanism of Alzheimer's disease for female. So maybe today I just recommend the 4 genes about the Alzheimer's disease for female, but maybe there is some specific mechanism of this kind of genes. Then we can find the relevant mechanism of target genes, and also we can study about the modulation of this target gene by using such as CRISPR-Cas9, the genetic modification, or some kind of gene, the knockout or gene knockdown, and the kind of things. Then maybe we can find the sex difference of the specific genes. Then maybe we can go to the personalized medicine for female for Alzheimer's disease.
Thank you.
Yeah.
Do we have any other questions?
I put another question in the chat too.
Oh, yeah.
Oh, yeah.
I can read the questions. I mean, what are some of the most critical gaps in our current understanding of sex differences in Alzheimer's disease? And how can future research address these gaps?
Gaps in our current understanding.
So it is a minor, very minor question, but in your slide, they're showing the effects of ApoE across the gender, sex. Like, I was wondering if the y-axis are in the same scale.
Yeah, I also curious about that actually. But I don't know, the y-axis is not here, but actually this is the research data. It is not the cutted data. So the paper did not show the y-axis about the female. Maybe I'm not sure it is same with this y-axis.
If it's same scale, because I saw that in age range, like after 60, or the female, there are more risks in female, but the scale shows
right,
an
exactly.
opposite. So I was
right. So I was also curious about that, but this paper just mentioned that the difference of the genetic factor of ApoE. But as you can see here, the immediate recall means the recall of their memory. But as you can see, if the y-axis is same with between the female and male, then it looks like the female have the good memory recall rather than male. So I also curious about the data, but this paper just announcing that the difference of the genetic, the factor of the ApoE between the sexes. So I think we need to check the data. I'm not sure.
Okay.
Yeah.
So in your opinion, what could be the most critical gaps in the sex differences in Alzheimer's disease?
Yeah.
In terms of brains and metabolism.
Yeah, maybe the sex hormone can make the big difference gap, because it is very, I think it's very difficult to study about the sex hormone in Alzheimer's disease, because basically the sex hormone is very have many the fluctuation, although the same within the woman, the sex hormone is very fluctuative, so maybe it can make the difficulties to study about the link between sex hormone and Alzheimer's disease. Maybe it can make some the big gaps to study about the Alzheimer's disease for female.
I see.
Yeah.
Okay, thank you very much. If we don't, do you have any more questions?
Hi, I have a question actually. Thank you. This was very informative. Congratulations, Dr. Park.
Thank you.
This was really great. I was wondering if in the US, you know, a lot of the research regarding women's health, we look at it in terms of segments, not just by age, but by different ethnic groups and races. And I was wondering if your research is very specific to South Korea, or if you're looking at it from sort of more of an like an overall, more of an overall look of, like, you know, looking specifically at, you know, Asian countries and what those differences are. And if you could elaborate on some of the groups that you were looking at, and if there was any differences.
Yeah. Actually, this database is our database is from not the Asian cohort. It is derived from the publicly database. It is derived from the maybe the Caucasian. So we couldn't see the difference between the Asian and Caucasian and some other the ethnic groups. So I really want to study about the difference between the ethnic. So but maybe it needs much higher the cohort and much the worldwide based the studies. So maybe please many clinicians and do these kind of researches for the many for the world. And however, about the blood-based biomarker data, we use the Asian participants, so maybe it can associate with the Asian group. But also, this is also the limitation about the it can also limitation because we did not perform this blood-based biomarker study by using other ethnics. But maybe I can do, because maybe there are many publicly available the blood-based biomarker data. So I'm trying to do this kind of analysis by using other the different ethnic groups. So maybe it will be the future research. So I hope many researchers have to do and should have to do this kind of the researches.
Yes.
Yeah.
Thank you.
Yeah, thank you very much.
Thank you.
Thank you. I think these differences in ethnicity, like differences in race and differences in socio-demographic status and differences in sex. I mean, this will naturally lead us to think about health disparity in research, which is our topic of the second talk.
Right.
So if there is no other questions, I'd like to move on to the next talk. Our next speaker is Dr. Hyo-kyung Grace Hong, and she is a senior investigator in biostatistics branch at the National Cancer Institute. And we'd like to thank our speakers for sharing their expertise and insight on this topic of why sex and gender matter in women's health research. And I also would like to thank the audience for the active participation. Please stay tuned for our next webinar, which is December 2nd. And we look forward to seeing you all in the future event. And have a great day and good evening to you all. Thank you.
Thank you.
Bye.